Yeah. Hi, good afternoon, everyone. My name is Vishal Joshi, and I'm the lead of automatic annotation in UniProt. So we are the team which is deploying the existing methods, prediction methods, evaluating new methods, and deploying them in production for annotating the unreviewed data set of UniProt KB, which is Tremble. So let's go right into it. I will be talking about the existing rule-based methods we have. And of recent, last year, we introduced ProtNLM, which is a natural language-based method for annotating protein names. Okay, so just a brief recap about UniProt. UniProt consists of all the databases you see on the screen. The blue one is the primary database, which is UniProt knowledge base, consisting of SwissProt as well as Tremble, SwissProt being the manually annotated set, while the Tremble is an unreviewed data set, but it is computationally predicted. When more than 95% of the sequences you see are coming from translation of coding sequences, namely ENN GeneBank, where UniPark is one of a comprehensive, non-redundant archive of sequences. So the sequences might exist in multiple databases. So UniPark will have one sequence and give one UPI ID, and then have extra cross-references to the multiple databases. And proteomes is the database which consists of a set of proteins exhibited in an organism, which consists of both Tremble and SwissProt, while UniRef is the clustered set of sequences at different resolutions. UniRef 90 being 90% similarity, UniRef 50, 50% similarity, and UniRef 100, 100% sequence similarity. So just before we go into how the predictions happen, let's talk about how manually and how automatically the annotation process goes through. So on the left-hand side, you see in the green box, it's a manual curation process where the curators will pick a set of sequences, run blasts on them, and check the homologs. Afterwards, they analyze those sequences by means of various tools where we have computational predictions for those sequences, and then
Sorry, we're not sharing slides online. That's why we got all these. I apologize.
No worries. Sorry for the interruption. So we were at the second step where the sequence analysis was happening, where the tools were going through the computational predictions and then analyzing which predictions can be merged into SwissProt. Then literature citations happen, where journal articles went through and then see the citations for those sequences in literature. Followed by, the fifth step is the fourth step, where the family-based curation happens. It happens where the reverse blast happens, where they find putative homologs and try to curate them as well. Followed by evidence. So they try to label the annotation with what evidence it comes from. For example, if it's come from curator or maybe inferred, all those evidence based on ecocode ontology. And then finally, the quality assurance happens where multiple curators review the annotations. So as you can see, it's a long-winded process and time-intensive and highly costly. That's why you see only around 570,000 proteins in SwissProt. While on the right-hand side, you see the automatic annotation process, where we use InterPro to get the set of signatures, which classify the protein entries by family, superfamily domains. Then for those set of entry signatures, we find what SwissProt entries match those InterPro signatures, which will act as a template. Followed by what annotations they have. For example, if they have consistency in keyword names, EC numbers, etc. And then we see the clades in the taxonomy to qualify certain annotations only for those taxonomies. And then finally, a rule is created. And then we apply rule at scale to the whole Tremble set. So for us, it's not only the accuracy of the method, it's also the scalability of the method that matters, because we have to apply it to around 250 million Tremble entries. Just before that, these are mutually helpful sort of processes, because the SwissProt is used for annotating Tremble, and when Tremble gets well annotated, based on the priorities, they get merged into SwissProt. So now we talk about an automatic annotation process, how the Tremble gets automatically annotated. The first rule we have, that's the first prediction method. It's a semi-automatic method where the learning happens through curators, and the application is computationally applied on the whole tremble set. So we have, these rules are devised and tested by the curators. So curators get these rules, and then there are around 8,335 rules, and these rules are applied by our highly scalable set of pipelines, and it uses state-of-the-art technologies like Apache Spark to combine to join the data and apply it at scale to the tremble data set. The unified rule system, as the name suggests, comes from a unity of multiple rules. So the rules comes from SIB, Swiss Institute of Bioinformatics, rule-based rules which come from EBI in UK, and the PIR name and site rules which come from PIR US. Now we talk about how a rule looks like. It's a simple if and then statement. So if you see on the left-hand side, the if part, there's a set of the conditions. If the rules match the condition, the right side, which is the annotations, will be transferred to that those set of entries. You can set the provenance of a rule through the rule ID, which is UR followed by the rule ID. But if the user wants to see which side the rules comes from, it is from the source ID. So if you see on the screen, it's source ID, which is RU something something, which is a rule-based rule. Similarly, if you want to see a HAMAP rule, it will be MF something something. So you can see the source of the rule as well. This is these signatures from UniProt glycosyl hydrolase signatures. So that's why you see on the right-hand side the annotations which give a protein name, similarity, and keywords matching those conditions. This is a very simple rule. The next rule you see is a complex rule where you have main conditions and case conditions. So you can have the main conditions will match, and then the annotations will be transferred. And then, for example, like we saw in the previous slide, the taxonomy checks happen. So if for fungi, the function is different. If the protein is not a fungi, then a different function is applied based on this rule. That's a more complicated rule. At present, the coverage with respect to 2303, which is the current public release, UniRule is covering around 34% of UniProt KB tremble entries, which is around 84 million entries. And on the right-hand side, you see the chart which tells you about how for each annotation type we are touching the entries. For example, keyword, protein description, keyword is around 67 million entries on tremble. So we talked about a semi-automatic system. Now Arba is a fully automatic system where the learning happens automatically, and so is the application. So Arba again learns from the experimental data in manual data set of UniProt KB, which is SwissProt, and it is based on frequency item set mining a priori. So it will find association of rules between the attributes, and based on certain quality measures in place, it will find all the rules which will define significant relations between the attributes and the functional information. Because Arba can create a lot of rules, most of the rules, sheerly due to feasibility, it cannot be applied directly. So it has to go through a rule reduction process, which I'll explain in the next slide. But then these rules are compared based on dominance and comparability, and it can do cross-validation, and finally it creates performance reports before it goes for prediction, which is the last step, where it applies those predictions for on the UniProt KB, again, tremble set. Arba is a multi-class system where it will, in a single learning process, it can learn functional information starting from protein names, metabolic pathways, EC numbers, all the way to gene ontology terms. Since the rules are quite large in number, sometimes there will be redundancy in the rules. So for example, one condition, a smaller rule will be touching a set of tremble entries, while a bigger rule is giving the same annotation type and value to those tremble entries. So either those rules are discarded, or if there is a significant overlap, these rules can be merged into one rule as well. And if you see Arba, the rule structure of Arba, it is very similar. It's following the UniRule XSD, but there's a subtle difference in Arba and UniRule rules. Arba will give only one annotation type and value, while UniRule will try to give as much annotations as it can. So it tries to annotate a tremble entry in depth. So consider it in computer science terms, just like horizontal scaling versus vertical scaling. So UniRule will try to annotate a full keyword, names, EC numbers, etc. Well, there will be multiple Arba rules which will be touching the single entry at different facets of information. The coverage of ARBA, because it's an automatic system, it has around 26,146 rules, which were applied in 2303, and it is covering around 50% of Tremble dataset. Now, before we go into Prot and LEM, we need to identify the problem we are trying to solve. So we want protein names, which are short and free text descriptions of a protein, and it will help to identify the common functions in a protein. For example, this protein is a capsid protein, which is a viral protein, and capsid is the protein which is the shell of the protein. The use case could be that user does a similarity scan, similarity search, and there would be a list of entries they would see, and these names will help identify common functions between all this set of entries which are as a result of the similarity search. And the requirement is to provide comprehensive and specific information about the protein, but be accurate, concise, and consistent for the names. This is the coverage you see on the screen, which is the UniProt KB coverage about protein names. So you see around there's a tiny portion of the names coming from SwissProt, but because the small data set is around 0.2%, while submitted names are more around 45% of the whole distribution. The red slice you see are Unitune and ARBA. So these names are coming from our in-house methods, which I explained earlier. And the yellow slice you see is one of the pipelines we have, which will give names to all proteins which have single domain. So the names which will be like X domain containing protein, they are those sort of names. Having all these systems in place, we still have the orange slice, which is 25% of the names which are uncharacterized. That means we can't give any meaningful name to those proteins. In the FASTA file, you will see with uncharacterized protein for those entries. In order to solve this problem, we collaborated with Google, and a method called Prot and LM, Protein Natural Language Model, was introduced. And this model is a text-to-text transformer model developed by Andrea Jane, Max, and Lucy Colwell Group. This will take a sequence of amino acid and will give a name, EC number, a family, etc. But we are using it for protein families at present. This was the training data set which was used. The data was based on UniProt 2021-02, and then it was tested on 21-03. So it was a temporal dev split where the names sequences which were new in 21-03, and the accessions which were not there earlier, on which that was the validation set. And finally, the test set was 2201, which consisted of all those sequences which were not in 2021-02, 2021-03, which was validation set. But in the test set, there was a challenging subset was identified. So we did not want to have those sequences which were very close to the training data set. So UNIREF 50 came into place, and all those. So we tried to find those sequences which were at most 50% similar to the training data set. So we wanted as far as we can, so we can see how the method generalizes to those sequences which do not have any similarity with the training set. So the model took a sequence and name pair to train, convert it into embeddings, and then uses encoder-decoder framework, and then finally gives a name as an output. The first model, Prot and LM, was introduced in 2204 in October last year. It was trained on only sequences and gave the names as an output, as you can see on the screen. We were having back then around 52 million uncharacterized protein, of which 49 million were given names in UniProt KB Tremble. However, in 2205, we identified that certain names are very closely related to the taxonomy they come from. So for example, for ovule protein names are usually found in plant-based taxonomies, while capsid protein is mostly found in viral proteins. So those sort of distinction required that taxonomy signal should also be supplied to the model. Yeah, and therefore there were 6 models deployed, 3 based on sequences and 3 based on sequence and taxonomy both. And then the ensemble approach will give a set of predictions, of which the highly scored one will be picked and applied for the Tremble dataset. In 2302, Google team found, especially Sergey, he found that structure could also be one of the information that could be given. That's why a 7th model was added. So we are yet to see the impact of this, but so far the impact is positive. So the mean accuracy score was around 0.3, but having this structure information, AlphaFold structures, secondary structures, it was around 0.84. So there was a minor improvement in terms of the score. These structure informations we saw that there are around 32 million uncharacterized protein, of which 24 million had structures. So we could use that information to apply. So for 2302, this model is in production in UniProt KB. We also identified that there were certain unmeaningful names which were slipped into the production, and most of the unsuitable names we found were semantically coming from submitted names. Therefore, we applied a quality check. We found the density of these unmeaningful names were below 0.2 confidence score from Prot-LLM. So that's why the threshold was applied, and many predictions were removed. That's why you see a drop in the names. So now only we apply. So now it is covering around 12% of the uncharacterized slice we saw. Because Prot-LLM is a deep learning model, it's sort of a black box. So in order to make sense out of it, Max has developed a pipeline, Max from Google DeepMind. So it will take the name, fetch all the sequences which has those names, and do structural sequence alignment to see which ones closely aligns. And then we can see that, oh, this is a corroboration from one of the UniProt KB dataset. There's a huge effort from manual corroboration as well from our curators who scan those sequences and find those names which cannot be applied. And then therefore we have developed a regular expression to remove these names as a post-processing of these names. So as an effect, you see on the right-hand side, the 25% names which were uncharacterized, 12% are covered now. So we have provided names for those 12%. We could have gone more, but since we are precision-oriented systems, we will try to have less coverage but have more meaningful names. You can find the Prot-LLM predictions with source Google. There are around 30 million entries which have these names. And the coverage, you can see the green chart, sorry, the orange line you will see is UniRule, which is more or less 30% covering the tremble, while RBA is around 50% when it was introduced in 2020. However, you see, after the introduction of Google, we are covering around 77% of UniProt KB in terms of the tremble coverage we have across the annotation types. And finally, these are a set of the pipelines we have. UniRule, RBA have explained, Prot-LLM have explained, and feature domains is the single domain proteins I talked about. We also have sequence enrichment methods, which using Phobius, Signal P, and which will give transmembrane predictions and morbidity light for disordered regions. And finally, on the right-hand side, you see a prediction file which goes into our production pipelines. For users, in order to apply these rule-based systems, we have Unifier. This is a Docker container where for this Docker container, you will give a sequence and UniRule and RBA rules can be applied. And then you can have the predictions from this. It will deploy the interpro scan to get the signatures as well before it can apply the rules. And the rule markup language is a bit well understood than UniRule XSD, which will tell you the conditions, the fact being the protein signatures, and the right-hand side you see the annotations which will be applied to those annotations to those tremble entries. With this release, we have protein sequence embeddings which were made live thanks to ROSLab Prot P5. We have per protein and per residue embeddings on FTP, and we provide embeddings on the fly through embeddings API. These are per protein embeddings. So yeah, thank you to the whole team of UniProt, to the entire UniProt consortium, all the collaborators, and thanks to DeepMind as well. Yeah.
Really interesting. I was wondering if you have a confidence metric that indicates how trustworthy each prediction is, or the rules are just binary.
So for the UniRule and RBA rules, we have a 95% criteria. So when a rule is made, it has to follow 95% confidence threshold before it can apply it. For RBA, it's even stricter. It is 100%. So the rules, when they find the annotation in the SwissProt templates, all the SwissProt templates has to match those annotations so that it can be applied. For Prot-LLM, it's a whole distribution from 0.0 to 1.0, but we apply from 0.2 and above. So 0.2 and below have more frequency of unmeaningful names. That's what we have.
And do you think if the rules are, well, maybe I'll ask you.
Yeah, sure. Okay.
I think you're done.
Thank you.